Do I have to hit anything, Bella? Okay. While my presentation's up, I'd like to thank ILSI and thank the organizers for inviting me to come today, and thank you for showing up at 8:30 in the morning on Monday. Okay. So this session is how much of a mystery remains with WGS. I think you heard a great start from Kathy, from Martin, and from Peter this morning. I'd just like to focus a little bit, a couple slides on, I guess, aspects that I hope are not a mystery. Starting off with, I think it's been wonderful, FDA, USDA, FSIS, CDC. So hopefully our raw data is not a mystery. So once again, if you have access to bioinformatics resources, you can go look at the raw data that the FDA, CDC, and USDA are producing. Admittedly, the metadata is limited. It doesn't have facility names, it doesn't have detailed, case information about patients, but that raw data, thousands or tens of thousands of isolates from the different agencies, is publicly available. The other point I'd like to make is our analysis methods are public. I think I've heard a couple of different comments that there are thousands of different bioinformatics pipelines, and that is true, but the FDA and the CDC have been very clear about which methods we are using. And we've done comparison studies between the FDA, CDC, and NCBI to show that our methods are equivalent, producing very similar results. Our method's up there, the CDC is up there. Additionally, Paul Keim, there's other competing pipelines out there that give very similar results for SNP analysis. The CGE and MLST, WGMLST methods are out there in bionumerics. Those are available. And, if anyone tells you that WGMLST is giving you a different answer than SNPs, that's not correct. They are giving just about the same answer, once again, for public health applications. Regarding the interpretation, I agree that sort of the global, recommendations or guides for interpretation, are still developing, but once again, there's multiple comments that, there's a lack of validation or unclear, lack of clarity about how reproducible the technology is. We've published a proficiency testing studies. You've heard a little earlier about the Global Microbial Identifier Group. So 2 different reports, including both wet lab and dry lab components, are available, on the Global Microbial Identifier site. I believe the 2015 study included 47 labs. So we looked at interlaboratory variation for WGS across a range of pathogens and a number of these labs. Those reports are available. Even though, once again, sort of the global guidance is not available, lots of retrospective outbreak studies, CDC, Public Health England have wonderful studies out there. I know from the FDA side and FSIS, we're working to publish, our sort of case studies around certain products or commodities or growing regions and putting that information out there. And so I hope those aspects are at least not a mystery or not as much of a mystery as they were. And so from my perspective, where does the mystery lie? And I think one sort of ongoing problem still, for us is that, it's not a mystery to bioinformaticists, but still WGS, it's largely not accessible to non-experts. And so we're working to develop easier tools. Bionumerics has made great strides, I think, the past couple of years to become more push button and easier to use. And that the data and results are very, I'd say accessible for bioinformaticists, people who are used to working in human genomics, but there's still a hurdle for people who aren't experts in that area. I'll also agree, there certainly is bias or potential for bias in the existing databases. Most of the samples are going to be clinical or of animal origin. That limits what we can do in terms of, if we don't have a lot of isolates that didn't make somebody sick, then it's difficult to do the comparison studies and actually, start to identify what determines pathogenicity or virulence and what those elements are. And the other point, I think, we hear a lot about sort of interpretation in the context of outbreaks, but in some sense, that the sort of typical case for the FDA, and I think certainly for industry too, is there's not any clinical findings. So we may have environmental positives, we may have food positives, we may have positives from a raw ingredient. And so interpretation of those findings sort of in the absence of an outbreak where we just may have environmental data, I think I agree that that is still developing even though we have a lot of information out there and we're trying to publish that. I think the other aspect that, is still trying to develop, I would say, once again, WGS is great for outbreaks, but if I'm industry, how do I use it to improve food safety? So I think we're working to put together, better functional databases. So if you do get a finding, it's not so much, from a food safety perspective, is this a match or not a match to a clinical isolate, but maybe, does this have resistance to cleaners? Does this have certain biofilm properties that I need to be more or less concerned about? And putting those sort of tools and databases out there for people to use. Okay, that's it. Thank you.